Fighting. Yeah. Are you science scout? I saw you earlier. Your brain must be full. It's been a busy day. I know. I was like, I haven't had a chance to like. Not like a blind board and playing around and then change how we got last night if you're setting the stage. But I'm not afraid of
All right, good afternoon, everybody. We're right at 4 o'clock. Go ahead and get started with our Good News Thursday. I got an email from Anna saying that she was awarded the John D. Vise International Discovery Fellowship, and so now she will be studying integrative cardiovascular control course in Copenhagen, Denmark. And then she also reminded me to say that last week we had 2 very successful days at Hickman High School where we did some outreach to the high school students, their exercise physiology course, and that was amidst a tornado also. So we were forced to the basement. So thanks for everyone that contributed.
Yeah, good for you. Okay. Hey, welcome everybody. I'm really excited about this talk because this is something that, few of us in the nutrition department work in this area. So it's my pleasure to introduce Dr. Tiffany Weir, and she is a researcher and professor in the Department of Food Science and Human Nutrition at Colorado State University. She has a bachelor's in microbiology and then she followed that up with a master's in plant pathology and then a PhD in cell molecular biology from Colorado State. So she's established herself as a leading expert in the intersection of diet, gut microbiome, and chronic disease prevention. In fact, I was telling my group today that we're working on a systematic review that's looking at the effects of dietary intervention on the gut microbiome in individuals at risk for colorectal cancer. And I didn't realize she was one of the last authors on 2 of the papers that actually made it to the systematic review. And I was very excitedly telling my group that, and my undergrad, whose eyes lit up because she recognized that paper. She's like, really? so yes, her team's work is having an impact on researchers across the US and internationally in this field. So she does really groundbreaking research integrating gut microbiome, molecular and metabolic data sets, those large-scale data sets that you collect from these studies, to explore how diet microbiome interactions influence conditions such as colorectal cancer and cardiovascular disease. So her work is highly interdisciplinary. So she combines clinical trials, preclinical studies, and advanced microbial ecology techniques, as well as omics, to understand those mechanistic relationships between the microbiome, diet, and vascular dysfunction. She's published over 70 peer-reviewed papers in high-impact journals, including Science, Nature Microbiology, and Cell, Host, and Microbe. And her research has been supported by federal agencies like National Institutes of Health, USDA, also the American Heart Association. And this one's interesting, Institute for Cannabis Research as well. I would love to read some of that research. So beyond her research, she serves as the director of graduate programs for the Department of Food Science and Human Nutrition. And she teaches courses on fermentation microbiology and phytochemicals and probiotics for health. And her teaching methods emphasize interactive learning and real-world application, something a lot of us in the nutrition and exercise field extremely value. She has received numerous accolades, the most recent ones including the College of Health and Human Sciences Award for Scholarly Excellence and the Best Scientific Contribution Award at the World Polyphenols Application Congress. And her work continues to shape the future of microbiome-targeted therapies, making her a leading voice in this field. And please, let's just give her a really warm welcome.
Okay. Is my mic turned on? Can you all hear me? Okay, I may not need it if you can hear me. First of all, thank you for the lovely introduction. And I've had an amazing day. I've gotten to talk to, I think, a good number of you in this room, and it's been really interesting and stimulating conversation. So thank you for all the time that you've given. I know what a busy time in the semester it is for everybody, and I've felt very welcomed here. So I will go ahead and sort of jump right in. I do want to throw out the caveat. You mentioned that I do very interdisciplinary work. And so that means that I am not alone in doing this work. I have many collaborators. So some of the stuff that I'm going to talk about today, I'm not necessarily the lead cardiovascular person. So if you're going to have a bunch of questions about the cardiovascular physiology, I will do my best. But I am the microbiota person on the cardiovascular project. So with that, we will go ahead and start. Okay. There we go. Okay. So just a brief introduction to cardiovascular disease. I don't think it's news to any of you that cardiovascular disease is one of the leading causes of death in the United States, as well as is becoming the leading cause of death worldwide. And this includes both in men and women. So what our research group is interested in is prior to the development of cardiovascular disease, there are some preclinical manifestations that occur. And the one that we are most interested in is termed vascular dysfunction. And vascular dysfunction is characterized by two different manifestations. One of these is arterial stiffness, and the other one is endothelial function. And I'll talk a little bit about both of these today, how we measure these, and what the implications are for these. But both of these conditions are strong future predictors of cardiovascular events. And so the nice thing about working in vascular dysfunction, being in a nutrition field, is oftentimes when you already have overt disease, it's going to be really hard to do something like a nutrition intervention to try to reverse that. But when you're doing something like vascular dysfunction, you haven't gotten yet to that stage of disease. So it's a really nice time to be able to go in and do interventions that are related to exercise, to diet, dietary supplements and things like that. So the first of the two things that I mentioned is arterial stiffness. And arterial stiffness is exactly what it sounds, right? It's basically reduced elasticity of the arteries, which makes them less compliant, and then they become more resistant to blood flow. So increased stiffness essentially is going to put a strain on your heart. It increases your pulse pressure and it makes your heart work harder to pump blood. And so you have increased risk of developing heart attacks and things like that. It's also associated with increased atherosclerosis development. And the way that we measure arterial stiffness, I work in both preclinical and clinical models. And so jumping way ahead here. So the way that we measure this is called pulse wave velocity. And we can do the exact same technique in both animals and in humans. Essentially what we do is we put Doppler probes on two different points. It's different in humans. We do the carotid to the femoral artery. When we do it in mice, we do it on the aortic arch to the abdominal aorta. And then you measure the distance between those 2 points, and you can measure the rate of the pulse wave that flows from that using the Doppler, and then divide it by the distance. And that's how you're able to get a measure of arterial stiffness. And so essentially a lower pulse wave velocity score, like the 350 centimeters per second, is a good good arterial stiffness. That means that you have compliant arteries. And if it's that 500 centimeters per second, that would be considered a high pulse wave. And so that's considered arterial stiffness or dysfunction. The other aspect of this is endothelial function, and the endothelium is the single layer of cells that lines the inside of the blood vessels. Endothelial function is determined by the ability of the vessels to constrict and dilate in response to both physical and chemical stimuli. And so we have a couple techniques for measuring this as well. In animals, this is a terminal technique. And so essentially we have to sacrifice the animals, and then we take either the aorta or we can take mesenteric arteries, and we mount them on probes on a machine that's called a pressure myograph. And we keep those arteries physiologically alive. So we have them in a buffer so they remain alive. And then you can pre-constrict them with some chemical constrictors and then add acetylcholine in serial doses, and that causes the dilation. And then you actually have a microscope on that so that you can physically measure the amount of dilation that you're getting. And so you can see how much those arteries are able to dilate. In humans, this is done also visually. You can do this with an ultrasound. The gold standard for this is what's called flow-mediated dilation. This is more of a physical stimuli because you put a blood pressure cuff on the arm to occlude the blood flow for a period of time. And then when you release that, and that blood flows. The shear stress then causes the release of nitric oxide and allows for the vasodilation to occur. And again, with the Doppler, you can actually physically measure that. There is another technique that we also use. I'm not going to talk about it in any of the research that we did today, but it's called the endopat. And it's essentially you put a probe on each one of your fingers. You do the occlusion and reperfusion again, and you measure the difference in the blood flow between the arm that was occluded and the arm that wasn't occluded. And it generates an index that's called the reactive hyperemia index. And then there's kind of a baseline level that if you're below that baseline, that's considered to be endothelial dysfunction. Okay, and so the last part of the intro here is where I'm a little more comfortable, is that's in the gut microbiota. And the gut microbiota, I think probably at this point everybody's fairly familiar with, but it's the trillions of microorganisms that reside within the human gastrointestinal tract. And these are inextricably linked to our natural function. And so we have evolved with these microorganisms. And as a result, a lot of the energy that we extract from the foods that we eat actually are coming from metabolites that are produced during the fermentation of complex carbohydrates and fibers and things like that by the gut microbiota. So they're really important in our digestion. They produce proteases and enzymes that our bodies don't, so help us break down proteins and peptides. It's a little bit less understood how it works, but they're also involved in fat metabolism and absorption of fats. And they are important for development and maintenance of immunity. There have been several studies that show that if you have aberrant colonization events early in life, that's been linked to the development of autoimmune disorders, food allergies, asthma, and things like that. And then also as we go to the other end and start looking later in life, decreases in certain types of microorganisms are associated with immunosenescence. So particularly like loss of bifidobacterium in your natural gut populations as you age is associated with reduced ability to respond to infections and things like that. And then the last one is pathogen protection. And so every day we touch surfaces, we eat food, we put things in our mouth, and we are constantly encountering things that could potentially become enteric pathogens, but we don't often become sick from these. The reason that we don't is because we have colonization resistance that's established by our gut microbiota. They produce natural antimicrobial compounds. They're better at taking up the substrates, which basically starve off any potential pathogens. They take up all the good real estate, so there's nowhere that pathogenic organisms are able to really attach, and a lot of them need to have an attachment in order to start stimulating, expression of pathogen-associated genes. And so without our gut microbes, we basically wouldn't be able to survive. I've spent about the last 10 years looking at the impact of the gut microbiota on cardiovascular disease. And the way that the gut microbiota can impact cardiovascular disease is through several different mechanisms. One is that through moderation of inflammatory responses. And so I hate to use this term, but a lot of people are familiar with it. So I'll throw it out there, but leaky gut, right? So there's this whole idea that if you have a disruption in your gut microbiota, you start to get a degradation of that mucus layer within your intestines that protects you. You start to get inflammation in those intestinal cells, and now things can move between the tight junctions and get into systemic circulation that are normally contained in the lumen. When that happens, those molecules that are able to translocate into systemic circulation can generate inflammation in other tissues and organs of the body. And so that's why the gut microbiota is associated with the development of things like diabetes, cardiovascular disease, even different mental health disorders, because it can generate this inflammation in locations outside of the gut. Immune modulation, we already talked a little bit about that. It's important in early life, it's important in later life, but ultimately it is important in moderating responses throughout our life. Our 80%, I've heard this number thrown out, I don't know if it's true, but 80% of our immune system basically is in our gut. And as we have over 13 trillion microorganisms that live in the gut, then they're a very important system of sort of moderating and training that immune system. And then the last way is microbial metabolites. Probably the most well-known example of this is the trimethylamine story. So choline and carnitine, which you encounter in the diet, can be converted by certain gut microbes into a compound that's called trimethylamine. trimethylamine then goes through the liver where it gets converted into trimethylamine oxide. And high circulating levels of trimethylamine oxide are associated with reductions in reverse cholesterol transport and the development of atherosclerosis. This is, a huge field of work from Stanley Hazen's group at the Cleveland Clinic. They've basically worked out a lot of those pathways, and they've also recently identified some other microbial metabolites that they think are contributing to the development of cardiovascular disease as well. So I mentioned that I have spent the last 10 years looking at the relationship between the gut microbiota and cardiovascular disease. So I'll go through some of our earlier studies. We, again, have always sort of focused on the vascular dysfunction, so that preclinical manifestation of cardiovascular disease. Oh yeah, there we go. Okay, so I'll talk you through this. So this was a study that we did in diet-induced obese mice. And we had animals that were on a standard diet, animals on a Western diet that we fed them for 5 months. The nice thing about the pulse wave velocity technique is that it's non-terminal, so we can do this multiple times throughout the study. So this is the pulse wave velocity, and you can see all the groups of animals were at the same point at the start. And then after 5 months, the animals that were on the Western diet had developed diet-induced obesity, but they had also developed arterial stiffness. So at this point, at 5 months, we took half of the animals on the Western diet and we gave them antibiotics. And this was not recommended at all, 100%, like wouldn't work for humans. But we gave them a pretty serious cocktail of antibiotics in their water every day for 2 months. We did quantitative PCR. We basically completely wiped out their microbiomes. They were essentially germ-free animals at that point. But what we found when we went back at 7 months and looked at the pulse wave velocity again was that the animals who were given the antibiotics, their pulse wave velocity values had returned to that trajectory that occurs naturally with the mice aging. So you can see it goes up here gradually as the mice get older. But the animals that were on the Western diet that didn't receive antibiotics continued on that higher trajectory and had much higher pulse wave velocity. So basically, we were able to completely reverse arterial stiffening by introducing the antibiotics. Now, the other thing that we did was that technique to look at endothelial function. And so we terminated the animals, excised their vessels, put them on the pressure myograph, and then looked to see what their responses were to acetylcholine. And essentially, we see the same thing. The animals on the standard diet had really good dilation at the end of the study. The animals on the Western diet were down to only about 60% ability to dilate. And then these animals that received the antibiotics were not statistically different than the animals that were on the standard diet. And then down here, you can just see this is those curves expressed as an area under the curve. So obviously, it's not very feasible for humans to just take antibiotics for the rest of their life and suppress their microbiome in order to enhance their heart health. So we wanted to look and see, like, well, can we identify any more about whether the microbiome is actually driving this and how the microbiome is driving this? So in this case, we used a model that was a genetic obese model for mice. So these were OB leptin deficient animals, and we also had control animals. And what we did is we had one cohort that we sacked and we collected their fecal contents. And then we had a second cohort that we gave antibiotics for 2 weeks in order to suppress their gut microbiota. And then we did these transplantations with the fecal contents that we had collected. And so we had control animals that received a microbiota from other control animals. We had control animals that received the microbiota from obese animals. We had obese animals that got a control microbiota and obese animals that got an obese microbiota. And what we found is here's the control, their pulse wave velocity. So they had nice compliant arteries. But the animals that were control animals and got the obese microbiota actually start to show some dysfunction here. They hadn't developed dysfunction yet to the point of the OB mice, but they had developed more dysfunction than the control. And that was just from microbiome transplantation alone. They were all on the same diets. Nothing else was different, only the microbiotas that they had received. Now, interestingly, we weren't able to rescue the phenotype of the OB mice by giving them the control microbiota. Some of this could be because the arterial stiffness, a lot of this, and we showed over here, is due to deposition of additional collagen. And so it's a lot harder to reverse the collagen than it is for that collagen to start to build up. So you can see there was increased collagen in the control OB animals. So this gave us kind of our second clue that the microbiota by itself is a factor that can drive the development of arterial stiffness. So again, though, these are all mice. And we wanted to know, is this actually relevant at all to humans? Because I don't know about the rest of you, but I spend a lot of time and money actually trying to kill mice in my house, not trying to prevent them from getting diseases. And so we really want to know if this is going to work for humans. And so we had a cohort of humans who we did measurements to find ones that had increased endothelial dysfunction. We specifically recruited lean individuals and obese individuals, and then we selected 2 individuals that were female and 2 individuals that were male that fell into the lean obese categories, but also had normal versus endothelial dysfunction as measured by the endopat. And we took the fecal material from these individuals, we transplanted them into germ-free mice, and then we just left the mice alone. We gave them a regular standard chow diet, no high-fat diet, and just waited for 8 weeks to see what would happen at the end of 8 weeks. And at the end of 8 weeks, when we looked at the pulse wave velocity, the animals that got, keeps doing it by itself here. The animals that got the lean microbiota were here, and the animals that got the obese microbiota had a higher pulse wave velocity. So again, signs of arterial stiffness just from the microbiota transplant. No other factors from these animals were different. We also had a cohort of germ-free mice over here for comparison. Germ-free mice would have been similar to the antibiotic mice in our other study, and you can see that the germ-free mice have normal function. This was with the 2 female donors, so we actually failed to reach statistical significance, but we did have a statistical trend. And then over here, this was with the 2 male donors, and we did have a significant increase in pulse wave velocity in the mice who received the OB dysfunctional microbiota. And then down here, the endothelial dependent dilation, we see similar things where the germ-free mice had the best dilation. Our lean donors, and this was with the 2 studies, we combined all these for the endothelial function, we didn't really actually have great. So just having a microbiota by itself had a negative impact on their endothelial function. But you can see the obese mice down here had really, really poor dilation. And then again, you can see this with the area under the curve. So we established a couple of things here. One, we hope that this is relevant to humans because even though we were still working in a mice model, we were working with a human microbiota. And 2, there is a clear causal role of the microbiota in the development of vascular dysfunction because all other factors the same, we saw differential responses when only the microbiota was being manipulated. So this has led to our next set of studies, which is, okay, well, we're not going to give people antibiotics. We can't make people germ-free. So what can we do? Are there some dietary interventions? And I work, these 2 studies, one is in a preclinical model, so intermittent fasting, and then the other one is in a human clinical trial with the blueberries. I work with Dr. Chris Gentile's lab on the preclinical and Dr. Sarah Johnson on the clinical trial for this. So I'm going to go through these 2 studies, basically looking at how either an intermittent fast, and in this case, I'm defining intermittent fast as a single 24-hour fast once per week. And in the animal study, we did this for 12 weeks, or how a 12-week blueberry intervention impacts vascular function and whether that's occurring through manipulation of the gut microbiota. I think I'm going to probably skip through this, but the reason that we looked at intermittent fasting in the first place is because there are quite a few different methods for intermittent fasting, but generally speaking, it's considered to be a bit more accessible to most people than standard diets because there are ways that you can fit intermittent fasting into your lifestyle. You could do a time-restricted eating protocol where you only restrict your eating to a certain period during the day. There's modified fasting where you reduce your calories during a fast day. So not necessarily every day, but you might have 1 or 2 days a week where you reduce your calorie intake to like 500 calories a day. Or you can do a strict, water only fast. The most popular version of this is what's called a 5-2 fast, where you have either 2 consecutive or 2 non-consecutive days a week where you fast and refrain from consuming all food during those periods. There is quite a bit of research out there that shows that these different modes of intermittent fasting have a beneficial impact on cardiovascular health. And that's kind of what's summarized with some of the studies in these tables here. Skipped one. So we did this study in animals. The 5-2 fast is pretty popular and fairly effective, but one of the issues with a 5-2 fast is that after about 6 weeks, they've done some studies where they look at compliance to the 5-2 fast, and people start to drop off. They can't maintain this for longer periods of time. And so we wondered whether if it was cut back to just one single 24-hour fasting period per week, if that might have the same benefits as doing a 5-2, and if it might be a little bit more accessible to individuals. Now, caveat here, these were in mice. We know mice metabolism is not exactly the same as humans, so a 24-hour period without food for mice is not necessarily going to translate to a 24-hour period without food for humans. But you work with what you have. So this is where we started. We want to bring this study into humans. So again, we used our OB mouse model for these intermittent fasting studies, and we did this single 24-hour fast for a period of, it looks like only 11 weeks, sorry, not 12 weeks. So we did this for 11 weeks, and what we saw is that the fasting didn't result in any significant changes to body weight. Now, this is important because if we had seen weight loss over this 12-week period, everything else I'm about to show you, people could say, well, the animals lost weight. That's why they had improved vascular function. So all the results that we see in terms of improved vascular function are independent of body weight. Down here just shows food intake. We did see like a decrease in the food intake in the OB animals over time. That's pretty common in that model. And then pretty steady food intake. So the fasted animals, there's this idea that if you fast for a period of time, you actually make up for those calories during the non-fasting period. That appears to be true because total caloric intake was not less, despite the fact that they were deprived of food for that 24 hours. And then we looked at the glucose responses, and we did see that, oh man, I don't have my key on here, but basically we did see that there was a beneficial glucose response in the intermittent fasted groups, whether they were wild type or whether they were OB mice. So we did see improved glucose curves in both of those animals compared to their respective controls. So the OB animals on the fast didn't have better glucose response than the wild type animals, but the wild type fasted had a better glucose response than the ad lib, and the OB fasted had a better response than the OB ad lib animals. And then when we looked at pulse wave velocity, again, we did serial measurements of this because it's a non-terminal. So we have 1 week pulse wave velocity, then we have weeks 5 and 6. The reason it's 5 and 6 is because it was a lot of animals, so we couldn't do them all in 1 week. But essentially, that's all one time point right there. And then we did a 9-week pulse wave velocity. And what we saw is that in the wild type fasted group, there were no improvements in arterial stiffness. And again, these are healthy animals generally on a standard diet. And so we didn't necessarily expect that there would be additional benefits with the fasting. However, in the obese animals, we did see improvements in their pulse wave velocity. And you can see this over here. This is our OB intermittent fasting. It was an intermediate effect. So it wasn't as profound, for instance, as when we gave antibiotics and we saw a complete reversal of the arterial stiffness. It wasn't statistically different from the obese ad lib, and it wasn't statistically different from the wild type. So kind of a little bit of an intermediate protection. And this was at both weeks 5 and 6 and weeks 9. So theoretically, 5 weeks of fasting was sufficient to start to see these beneficial vascular effects. That's another question people have about fasting, is what is the duration of periods of fasting that you have to see an effect? And then another question that we'd like to eventually answer in human populations is, once you start to see benefits, how long do you have to maintain fasting in order for those benefits to persist? So can you do a period of like 6 weeks of fasting, start to see benefits, go back to your normal eating for 6 weeks, and then are you going to maintain those benefits? And then could you go back to another period of fasting to kind of do like a maintenance so that you wouldn't have to say, okay, I'm going to fast every Monday for the rest of my life. So we were interested in what was the role of the gut microbiota, because we've already established that the microbiota by itself is able to impact vascular function. And there are several studies, because they're in, human and animal studies, where they've done intermittent fasting. And as you would expect, when you starve the gut microbiota, that's going to have an impact on that microbial population. And so we wanted to look and see what was the impact on our microbial population. I'm only showing you the OB animals here because the differences were much more pronounced in the OB animals than they were in the wild type animals. But you can see here that we have a pretty good separation between the OB intermittent fasting and the OB ad lib animals when we look at the beta diversity. And then you can see over here, as you plot this, primarily along that second principal coordinate axis, we get that separation. We have statistically different clustering here with the OB ad lib and the OB fasting. So the next question is, we expected that we were going to see changes to the gut microbiota, but what does all of this really mean? And we had clinical data, we had our pulse wave velocity, we had the microbiome data, we collected different tissues, and we profiled immune cells. We had PBMCs, and we're looking at cytokines and things like that. When you have these massive amounts of data, sometimes it's really hard to be able to draw these conclusions. And so we found a really nice bioinformatics program. It's called Volare. And Volare, I tried to remember earlier what the acronym stands for, but essentially you can put in all of these large data sets and it will do these multiple regressions and generate what are called top tables. And these top tables then you can use to start to look at relationships. And so we did these top tables with all of our different data sets, and we started to figure out, okay, where were these different relationships? And first, we wanted to show that there was no relationship between the change in pulse wave velocity and change in body weight, because it's something that reviewers always come back and say, like at the terminal point, you had that little drop. Are you sure that's not driving all your stuff? So there was no correlation between the body weight and the change in pulse wave velocity. So we're pretty sure that that was independent. What we did see is that there was a high correlation between the change in pulse wave velocity and several of these chemokines, in particular, PBMC levels of the CCL2 and the CXCL1. So then the next thing that we looked at was whether there was a relationship between these different chemokines and some of the different gut microbiota. And there's one that really stood out to us here. It's this Clostridia UCG14. That had a positive relationship with most of these chemokines that we measured. And then this other one, Mirabaculaceae, that had a negative relationship. Now, we're not as interested in the Mirabaculaceae because this was a rodent model, and this is primarily found in mice. It's not actually a very important component in the human microbiota. But this Clostridia, we don't know specifically if this one would be found in humans, but Clostridia are common in the human gut. And so this one might have more relevance. So we took all the data from our top tables and we decided to run a mediation analysis. And with the mediation analysis, what we found was this Clostridia UCG14 is basically mediating the levels of the CXCL1 and the CCL2 that we saw in the bloodstream, and that is what's having the downstream effect on pulse wave velocity. So we wanted to follow up with that. We went into some of our different tissues to look and see if there was elevated levels of CCL2 in areas besides the PBMCs. And what we found is that we had elevated levels of CCL2 in all of the adipose tissue. And we also did some histology, and we decided to look at the number of lymphocytes that were in the PVAT, because that's directly surrounding the vasculature, right? And so inflammatory molecules that are going to be produced by the PVAT are going to have a direct effect on vascular function. And what we found was that there were higher levels of lymphocytes in the PVAT of the OB ad lib compared to the OB intermittent fasting animals. So the current working hypothesis is that fasting is modifying the gut microbiota. The gut microbiota then is having an impact on the expression of these levels of CCL2, and that the CCL2 is a chemokine. So that's a trafficking signal, right? Which recruits immune cells to certain tissues. So when we have CCL2 expression in the adipose tissue, that's now recruiting these immune cells to the adipose tissue, generating inflammation in that perivascular adipose tissue that then is leading to the development of vascular dysfunction. We got some mice in about 2 weeks ago, and we're testing that hypothesis on the CCL2 end of it by giving them anti-CCL2 antibodies, as well as injecting them with CCL2 to see if we can impact the immune cells in the adipose tissue and the development of vascular dysfunction in those animals.
There's potentially negative effect of fasting in this model.
No, this would be positive. Yeah, so we see decreased lymphocytes in the fast. Yeah, so the microbes are suppressed with fasting. Yeah.
Pulse wave velocity again at the raw data.
Right here. Yeah.
Okay, so it's the OB intermittent fasting that's having the beneficial effect.
Right. Yeah. So we're seeing a benefit with that once weekly fast for over a period of 12 weeks in terms of improved pulse wave velocity.
That's associated with that chemokine.
Less chemokine in that one. Yeah. So we're seeing more when, yeah. So that probably wasn't explained well. So the relationship is as there was an increase in that Clostridium, we see an increase in CCL2, an increase in lymphocytes in the perivascular adipose tissue. With fasting, we get a decrease in that particular microbe, a decrease in the chemokines, and a decrease in the lymphocytes. Sorry, that wasn't clear. Yeah. You know, the grad students actually do all this work, and so I'm not the best at explaining it, but. So the other aspect of this is with that Volare program, we were able to identify a number of different microorganisms that had relationships, and some of them weren't necessarily tied back to the relative abundance of those microbes. And so there were some things that the relative abundance of those microbes stayed the same with fasting or with ad lib feeding, but their relationship to pulse wave velocity changed, whether it was in a fasted state or in a fed state. And so this is just the slope here for the intermittent fasting. This is the slope for the ad lib. So basically we're looking at these microbes here, were all the ones that sort of had a significant relationship with the pulse wave velocity. And this is an example of a graph then that can be generated from each one of those that shows what that relationship is. And here you see the change in pulse wave is positively associated with a change in Lactospiraceae, which was one of the organisms over here on this graph, in ad lib, but it had no relationship with it in fasting. So as you see an increase in Lactospiraceae in the fasted state, it didn't impact pulse wave velocity. So we started to think, well, what does this mean? Well, the microbes, when you're not eating, they're not eating. So we're starving those microorganisms of food, or they're scavenging. So they're eating things like your mucins, and they're going to change their metabolism, which means that the metabolites that they are producing may be different in a fasted and fed state. And so what we hypothesize is going on here in this particular case is that in the ad lib state, there's some sort of substrate that they're using to produce a metabolite that has a negative effect on pulse wave velocity. So we see an increase in pulse wave velocity with increasing bacteria in the fed state. Whereas here, even though we can see some of the animals had a high level of that particular microorganism, it wasn't associated with that change in pulse wave velocity because now that substrate is absent. So those microbes are no longer making whatever that metabolite is. Now we have some where the relationship is opposite. We're having a lot harder time trying to explain that, but this is an area that we're hoping to follow up on as well to try to identify what some of those metabolites are that might be mediating that. So our conceptual model here is basically when we have the 24-hour intermittent fast, It has an impact directly on the gut microbiota, and this is in the background of obesity because we didn't see this with the ad lib animals, or the wild type animals. And that's changing both the structure. So in this case, that's that Clostridium that did change. We saw a decreased relative abundance with fasting and an increased relative abundance in the fed state. And that Clostridium there is somehow mediating the levels of CXCL1 and CCL2, driving inflammation in the adipose tissue and development of vascular dysfunction. But also, on the other side, we had all those microorganisms that were in that little box that I said had significant relationships with the vascular dysfunction, and we can't contribute that to changes in their relative abundance. And so we think that that's changing the function of those microorganisms, probably through changes to their metabolism, which then is resulting to changes in vascular function as well. So we're going to switch gears. So I still have some time. Okay. So any questions before I move into the next kind of case study? Yeah.
So for the work you're doing with fasting, are you doing it, are the stack days structured so that they're X amount of days from the fasting, or? Okay.
Yeah. And actually, this is important, and I didn't mention this, but we make sure that all the animals are sacked after a refeed, because we did an acute study, and I don't have time to show the data here from the acute study, but in the acute study, when we do it, you have an acute effect on pulse wave velocity, and then immediately after refeeding, it bounces back and you go back to sort of normal or abnormal vascular function. And so we made sure that all of these animals were post refeed so that we weren't just seeing that, like acute fast effect. And we also took into account light dark cycles, because I know with animal studies, a lot of people do fasting studies and take their food away during their cycle when they're sleeping anyway. And so you don't get really good data from that. So we kept all those things in mind when we developed this study. And we repeated it in 2 cohorts of animals, and we got identical results in both cohorts of animals, which is something too that doesn't get done very often. So, all right. Any other questions? Okay. All right. So this is complete switch gears, because now we're not talking about withholding food. We're talking about eating more of something that's good for you. We're going to talk about blueberries, and this is not in mice. This is in humans. And so this was a study that was run by my colleague, Dr. Sarah Johnson. She is interested in improving vascular function in postmenopausal women. And she's been working with blueberries for quite a while. There's been evidence in men, there's been evidence in other populations that blueberries do improve vascular function. There's some studies that show that blueberry improves blood pressure. And so that's why we went with blueberries for this study, but basically the same hypothesis, right? So blueberries are packed with polyphenols. You can have direct antioxidant effects that could be benefiting vascular function just from the components of the blueberries. But there is, you eat the blueberries, right? So they make it into your stomach. The polyphenols are very poorly bioavailable, and so very little of those polyphenols actually get into circulation. And so our hypothesis was that the gut microbiota was actually going to determine what the circulating levels of those polyphenols are, and that's going to be what sort of mediates your response to blueberries in terms of vascular function. So in this study, this, as I mentioned, was 12 weeks. Sarah's studies are designed to be, real world applicable. So 22 grams of a freeze-dried blueberry powder that was supplied by the U.S. High Bush Blueberry Council. And if you convert that into cups, that's 1 cup of blueberries per day. So that's an actual achievable dose that you could eat just from buying blueberries from the grocery store. And then they have a placebo group as well. I think it's like a maltodextrin powder with like weird dyes and flavorings and stuff to try to make it like look and smell like blueberries. Her population was estrogen deficient postmenopausal women with elevated blood pressure or stage 1 hypertension, 45 to 65 years old. They measured endothelial function using that flow-mediated dilation technique that I mentioned as the primary outcome. Also looked at blood pressure. There were no changes. I'm not showing that data here. oxidative stress mediated endothelial function. So this was a really cool study. I'm also not going to show the results from, but they infused vitamin C to see if the impact from the blueberry intervention was due to reductions in oxidative stress, and we're able to show that it is because of reduced oxidative stress, blueberry polyphenol metabolites in gut microbiota, and then they had several secondary cardiovascular outcomes like augmentation index and things like that that they looked at. So again, blueberries did improve the endothelial function using the flow-mediated dilation. They also adjusted for shear rate here. And you can see here was the flow-mediated dilation. In this case, the higher number is better, right? Because you're looking at the dilation. So after 12 weeks, the women who were on the blueberry had improved flow-mediated dilation, whereas the groups that were on the placebo had no change over that 12-week period. And let's see, and then I think this is just looking at the change. So this is the change in flow-mediated dilation in the blueberry versus the placebo. And you can see also that the change in flow-mediated dilation was significant. They measured total polyphenols that were present in the plasma. And unsurprisingly, they had higher levels of blueberry-associated polyphenols in people who ate blueberries compared to people who did not eat blueberries. They looked over here to see what the breakdown of those different polyphenols were, and you can see that there were a number of different ones, but a big one here was hippuric acid, which suggests that some of these blueberry polyphenols are being extensively metabolized by the gut microbiota, because hippuric acid is one of the very end metabolic products of polyphenol metabolism by gut microbes. So that got us interested in like, okay, a lot of these polyphenols are being metabolized, so is this potentially driving response to this intervention? But before we got there, we wanted to look and just see, did the blueberry itself in the entire population have an impact on the gut microbiota structure? It did not. We saw no differences in the individuals before they took the blueberry for 12 weeks and after they consumed blueberry for 12 weeks. And so the blueberry didn't really have any major impacts on the gut microbiota composition. But what we did do is look at these individual data points for the flow-mediated dilation between 0 and 12 weeks. And this blue one here is the blueberry group. The red ones here are the placebo group. And you can see that there were a number of people that had very good responses to blueberry, but there were also several people that had no or sometimes even negative responses to blueberry in terms of their flow-mediated dilation. So we took just this blueberry group here, and we split them up into what we called responders and non-responders. And then we went back and we said, what if we look at the gut microbiome of these responders compared to the non-responders? And when we look at the non-responders here, we see that there's no change over the course, there's no change over the course of the intervention in the non-responders. There's no change in the course of the intervention in the responders, but there are baseline differences between the responders and non-responders in terms of the total richness of their microbiota. Is there a way to make it stop going by itself? Probably not at this point, huh? Okay, I'm almost through. So anyways, there were some baseline differences, and this isn't the greatest figure here because when you have these ellipses, it sort of masks, but this was also reflected here. You see that there's a baseline difference between these two populations. So this made us think, are these baseline differences perhaps associated with their ability to respond and with the amounts of circulating metabolites that they found in their plasma? So we took these samples and we did a metagenome analysis. So we took these and did shotgun sequencing. This was done in the lab of my collaborator, Dr. Kelly Wrighton. And I wanted to mention some of these tools that she developed. The first one is DRAM. So DRAM takes multiple databases and they use de novo synthesis of their metagenomes. And then they use JAM with all of these different databases to get the annotations, and they're able to come up with these different metabolic pathways that are much more refined than if you just do individual searches of your data against eggnog and keg and things like that. So then we took these distilled and refined metagenomes, And we put them into another database that Kelly's group has developed, which is called CAMPER. And CAMPER is specifically curated annotations for microbial polyphenol enzymes and reactions. Now, caveat here, Kelly is an environmental microbiologist, so her database is built and trained on microbes that they found in the soil and in streams and things like that. It wasn't built for the human gut. So we weren't entirely sure if we were going to find our specific metabolisms in her database. So that was the first kind of proof of concept. Well, nope, sorry, I guess maybe I don't have that in there. But we wanted to see if it was trained, and we were able to find several of our metabolisms in Kelly's database. We are working to build a gut-specific camper now that is built off of metagenomes from the human gut so that we can have a more complete picture of the polyphenol metabolisms. So the idea here is that when we consume these polyphenols, a lot of times they're going to be conjugated with sugar molecules, and that's going to make them less bioavailable. But when those sugar molecules make it down into the gut, the microbes are going to utilize those for energy. So they can remove those sugar molecules. They have a lot more diversity in their beta glucosidase enzymes than humans have, and so they can break down a lot more types of sugar bonds. And so we wanted to see if there were differences in glycosylase enzymes between the responders and the non-responders. We didn't have a sequencing depth that was significant to really be able to get a great picture of this. And so we saw, a couple of the glycosylases look like maybe they were a little bit higher in the responders compared to the non-responders, but we didn't see anything significant here. So then the next question is, okay, so maybe they're all equally deglycosylating these molecules, making the parent polyphenols similarly bioavailable, but the metabolism of those polyphenols is such that they get degraded before they get absorbed in the gut if you have like a high polyphenol metabolism gut. And so that's going to reduce the circulating levels of these compounds. And so we wanted to see if we could find any of those pathways in DRAM. We know that there are anthocyanins, flavonols, hydroxybenzoic acids, and hydroxycinnamate acids. And all of these pathways here for these organisms are encoded in the CAMPER database. And so we expected that we would be able to find some of these. If you look here, these heat maps show all the pathways that were encoded by CAMPER in our human data set. And then over here, these were some of the CAMPER pathways, and it actually continues on beyond that because there's much more metabolisms in the CAMPER database that we didn't find in our human data set. Probably some of that is because we had a low sequencing depth for the metagenome, so we didn't have complete coverage, and some of that is probably because there are just so many more microorganisms in soil that aren't in the human gut that can metabolize those polyphenols. But one that we found that was significantly one that we found that was not significantly different between the groups is that protocatechuic acid pathway. Protocatechuic acid is one of the primary metabolites from the blueberry polyphenols that's attributed for some of those beneficial effects. So we weren't necessarily modifying that particular pathway in the responders and the non-responders. However, we did see that there was a flavonol degradation pathway to phenylacetic acid, and that pathway differed in presence and absence. So that pathway was present only in our responders, except for one responder didn't have it, and largely absent in our non-responders, with, again, only one of our non-responders having that pathway. And essentially that takes one of the flavonols that's in blueberry and can break it down into phenylacetic acid, fluoroglucanol, or hydroxyuranone. So we went back to, now this is looking at the plasma metabolites from the humans again. And when we look at the plasma metabolites from the humans again, we actually saw that there were higher levels of some of these flavonols that were present in the responders compared to the non-responders. Now, we have higher levels of these in the plasma of the humans, but we have higher levels of metabolism that would break these down in the gut of the humans. And so what does that indicate? To me, I think what that means is that they were better, they had higher levels of the flavonols that they were starting then, which they started to break down in the gut. So they upregulated, the microbes upregulated those pathways, but ultimately more of those were able to get into circulation as well, because we're looking at those two different compartments there with that. And when we look over here at the flavanol abundance, again, you can see these were higher in the responders than the non-responders. And when we start to look at specific compounds, what were those flavanols that were higher in the responders than the non-responders? It's myricidin and quercetin. Both of those have known beneficial effects on cardiovascular function. So conceptual model, again, just is that people have different baseline microbiotas, and those baseline microbiotas are going to respond differently to the dietary intervention. Our prediction is that those responses then to the dietary intervention are going to result in differences of circulating levels of beneficial polyphenols in the blood, which then is going to have an impact on vascular function. We're hoping to do a much bigger study with much deeper sequencing and a gut-specific camper database to be able to follow up on this question. So some of our next steps are, do certain microbes influence the chemokine expression to recruit immune cells to the vasculature and drive inflammatory responses that initiate or exacerbate vascular dysfunction? We're looking at that now in mice. How do changes in nutrient levels and compositions alter microbial metabolism and metabolites? Can we identify novel metabolites that exacerbate or prevent vascular dysfunction? As I said, the protocatechuic acid has largely been the focus of research in terms of the blueberries, because I think that's the primary active compound where the contributions of things like myricidin and quercetin have largely been ignored, but could be important drivers of responses. Does the baseline microbiome composition determine the bioavailability? And then can we use that baseline microbiota to predict who would be a responder or a non-responder to a dietary intervention? So to wrap up, the fasting studies, I forgot to point out, I have like clocks and blueberries in the top so you knew where we were and everything, but Dr. Chris Gentile runs the Integrative Cardiovascular Lab at Colorado State University. And then we share these two grad students. Elliot did a lot of the immune cell work, and Scott does most of the microbiota work. Janet developed the Valari program and helped us figure out how to use it. And Taru did a lot of the immune cell work as well. And then for the human study, Dr. Sarah Johnson is the PI on that study. This was her PhD student, Emily Wolf. She's now down at Pennington Biomedical Center. And then Dr. Kelly Wrighton developed the DRAM and Camper tools, and Bridget McGivern is the one that helped us use them. And then Bree Risk was my PhD student that did all the analysis with all the data that resulted from that. And knowledge funding and the rest of our labs. Questions?
So for your great talk.
Thanks. Yeah.
For the studies that are looking at vascular function from the exercise vessels of mice, you use acetylcholine as your primary indicator of vascular endothelial-mediated dilation. Have you looked at using insulin at all, insulin-mediated dilation, to be clinically relevant for your OB models, for example?
We have not. No, we've stuck pretty much with the acetylcholine. In fact, we initially did a fasting study with the OB mice, and then we decided that was not really a great model for those studies, but we'd already started there, so we had to kind of continue down that road. But moving forward, I think we're going to try to move away from the OB model, because they have already an aberrant microbiota. So at baseline, their microbiota is different than the wild type animals. But more importantly, they have really weird physiological aberrations. So for instance, they have reduced collagen, even when they have vascular dysfunction. They have a thinner intima media. They have longer crypt villi compared, typically with inflammation, you get shortening of the intestinal crypts and we get longer. So they're physiologically very weird. And so I think moving forward, we're going to move back into the diet-induced obesity models.
So the second study where you showed the effect of supplementing blueberries on vascular function. How about the effect of fiber? Because when you eat fresh blueberries, they also have resource.
Yes, and fiber absolutely could be playing a role because it is a whole freeze-dried blueberry powder. And so it's not an extract. There are studies where they've used blueberry extracts and they see beneficial effects with just the extracts. And so there is an impact of the phytochemicals, right? But in this case, because it's a whole blueberry, the fiber could absolutely be playing a role.
It wasn't fiber matched with the control.
I don't think so, no. Yeah, because we're not controlling the rest of their diets, right? So it's just the 1 cup of blueberries. And so to fiber match doesn't make a whole lot of sense if they're eating different diets in the background. So.
I'm just curious, in your mouse model, when you were looking at the antibiotics, so the antibiotics actually improve vascular function. I'm just curious, do you look at sex differences of the mice?
Those were all male. Yeah, we used all male. Yeah, so we used all male mice in that study, but the study where we had the germ-free mice that got the human transplants, we had male and female mice. The female mice will develop vascular dysfunction, but to a lesser degree than the male mice, especially because we're using younger mice, right? So we had a mix of male and female mice that got either the female microbiota or the male microbiota, but the group that had the female microbiota, we had slightly more female mice. And so there was a little bit, if I had colored them by mouse sex, I think what was driving that down was the female mice. And that's why we only got a statistical trend rather than actual statistical significance, is because they started to develop dysfunction, but they were a little bit more protected because of the estrogen, right? Yeah. We did not. We really just were interested in, more so we recruited based on lean and obese because people don't know what their vascular function is, with the hopes that we would find people in those 2 cohorts that had what we considered to be normal function and dysfunction. And so really it was the obese cohort that we were looking for, because you can have the metabolically healthy obese, particularly when you're working in a younger population. And I think that we didn't want to move into that territory where you start looking at age-related vascular dysfunction. And so we kept our enrollment age, I think, to 40 or 45 for that study. And so we recruited obese individuals in the hopes that some of those obese individuals would have that endopath score below the 1.67. And that was the criteria that we used for the endothelial dysfunction for the transplantation study.
One of our Zoom participants, Melinda Himmelgarn, host of Food Sleuth Radio, wants to know, have you been able to ID any protective gut microbiota that could be supplemented to protect against cardiovascular?
We've not.
We also had a second question. Is any of your federal research funding being cut, or will your research be?
We have, so the blueberry study, what we want to do with that, the gut DB, we actually have an RM1 that's under review, went to council on Tuesday, and I'm obsessively checking my cell phone to see if NIH has given us a message yet, if it's going to be funded or not. It was looking pretty good, but we don't know right now. I think, yeah, nothing has been cut, but a grant that should, was on the verge of funding may or may not be funded, and we're waiting to find out.
There's a study where at 12 weeks you started to see a fall in one of your outcomes. Was that a human study?
Remember, do you know what the outcome?
It's the effect of polyphenols with supplementation.
Oh, it was the metabolic. Yeah, that was actually really interesting. I don't know if people got study fatigue. Yeah, because we saw there was a peak in the polyphenols at the 8-week time point, and then it drops down lower at the 12-week.
Britt Burton was here talking about strawberries in a supplementation study, and she found exactly the same thing. Start to go down after 10 weeks. And I'm wondering if the human doesn't begin to adapt. So you're feeding the bugs a new thing, and then somehow there's an adaptation.
And that's actually a great question, because I said that the microbiome didn't necessarily, but we also showed in the fasting study that in absence of changes in relative abundance, we still saw changes in metabolism, right? And so we only had one time point for the metagenomics. We didn't have the 8 and the 12-week. We actually, to be fair, we had both, but not necessarily all for the same people. It was like whatever was left in the freezer, because this was an afterthought for the experiment. And so some of them were 8 and some of them were 12. Yeah, but that could be that there's either a metabolic adaptation where they're starting now to break those compounds down and there's less of it getting into the blood by 12 weeks. Or it could be participant fatigue at 12 weeks and that compliance starts to drop off. Of course, they don't report that and we show that they still maintain high levels of compliance, but.
Or a better clearance in usage so that the concentration is lower in the blood.
Right. Right, yeah, so a lot of different, that's interesting though, the report, yeah.
All right, let's thank Dr. Weir. Thank you.